We okay? All right. All right, moving on to the Krebs cycle then. What else could this cycle be called besides the Krebs cycle? Did anyone see that? What else the Krebs cycle could be called? Citric acid cycle. It's important that you know both. The AP test uses both interchangeably. So I don't know which one it's going to use this year. Okay. Where does the Krebs or the citric acid cycle occur in our cell? In the mitochondria. Where in the mitochondria? In the matrix. Good. Okay, so it's in the matrix of the mitochondria. Excellent. Now, what am I going to start with? What molecule am I going to start with in this Krebs cycle?
Acetyl-CoA.
Acetyl-CoA. Good. Acetyl-CoA is going to enter in. Now, acetyl-CoA is going to combine with this 4-carbon molecule. Remember, acetyl-CoA is a 2-carbon molecule, and I have to use my cheat sheet because I can never spell these. Okay, oxaloacetate is this 4-carbon molecule that my acetyl combines with. And when it combines with the oxaloacetate, it produces a 6-carbon molecule called citrate. Now, which one of those has higher energy, oxaloacetate or citrate? Which one's a higher energy molecule based on your activity yesterday? Citrate. Why is citrate a higher energy molecule than oxaloacetate? What makes this have higher energy than this one? Anyone?
Does it have more potential energy?
Has a greater potential energy because this is the molecule that we form in the end, right? After all of the energy's been removed throughout this cycle, and we'll see that energy in a minute, but as it's been removed, then this is left. So where's the citrate getting this potential energy from? What's being combined with the citrate to give it more potential energy than oxaloacetate? Yeah, say it. The acetyl. Right? So the acetyl is binding with this oxaloacetate, making this a higher potential energy molecule. Now, there are a lot of different molecules that go along this process. The only two you are expected to know, oxaloacetate and citrate. Acetyl going in. Okay? So I'm going to leave the names out, even though I'm going to be like drawing them, but I'm going to leave the names out of all these other molecules. Okay? So from citrate, we are going to form a 5-carbon molecule. If we formed a 5-carbon molecule, what do you think was removed? Carbon dioxide. And if I told you that this was an oxidation, or citrate was oxidized down to this molecule, what else do you think is happening? NAD plus is being reduced to NADH. Good. This is going to happen again. And I'm going to end up with a 4-carbon molecule as CO2 is removed and NAD plus goes to NADH. Okay? Now, you might think, well, this is a 4-carbon, this is a 4-carbon, what's going on in between? This is called acetyl-CoA, and it goes through a series of reactions before it goes back to oxaloacetate. part of that is taking ADP and inorganic phosphate and converting that to make what? ATP. So we're getting actual ATP energy from this. We have another redox reaction happening where the succinyl is being oxidized to oxaloacetate. If that's happening, what do you think is what these processes occurring? NAD plus to NADH. And we have a second reaction happening here where FAD is going to FADH2. Now we're back to that oxaloacetate, to that beginning piece. Now, how many acetyls did we have per glucose? How many acetyl-CoAs did we produce per glucose? 2. So how many times does the citric acid or Krebs cycle happen per glucose? 2. So let's tally it up and let's do a net production. My net per glucose. How many ATPs am I going to form in the Krebs cycle through, or per glucose? 2. Okay. How many NADHs am I going to form per glucose? 3 per Krebs, 2 cycles, so 6. How many FADH2s are going to form? 2. And how many carbon dioxides are going to form? 4. Good. So now let's talk about where are these going. Where is the ATP going to go? It's going to be used by the cell. Good. Okay. Where is the NADH going to go? Oxidative phosphorylation, or ETC. Okay. Where's the FADH2 going to go? Is FADH2, how was that formed? Through? We went from FAD to FADH2. What happened during that process? Electrons were added, right? And so it still was that reduction reaction. And so do you think that this has extra electrons on it? Yeah. Where have all of our extra electrons gone so far? To oxidative phosphorylation. So this is going there too. Okay. And where's our CO2 going to go? Exhale. Good. All right. So now let's come over to our summary. I need my purple marker. How many ATPs are formed from the Krebs here? 2. How many NADHs? 6 NADH and 2 FADH2. And we said that those were going to be going to our last step into the ETC. Anything else from Krebs? The one thing I'm missing on here is that Lynx has CO2 and Krebs has CO2. I didn't add those in, but you can add those if you want. So now we're going to our last step, and our last step happens right here, right on this inner membrane of the mitochondria. And we call that oxidative phosphorylation. And I'll write that whole thing out so you don't have to, I didn't write it all up here. My markers down. Did I spell that right? I don't think so. Phos4Y. Relation. Alright. So we keep calling this the ETC. Really, this has two parts of it. It has the ETC, or the electron transport chain. And hemiosmosis as part of it. And where did I say this happens? The inner membrane of the mitochondria. Okay, awesome. So it's on the inner membrane of the mitochondria. So I'm going to draw this out. This is my outer membrane. This is my inner membrane. This is the inner membrane space. And then this is the matrix of the mitochondria. Okay? Within my inner membrane, I'm going to have a series of proteins. So protein channels that are going to be within my inner membrane here. Okay? So what's going to happen is all of these NADHs and FADH2s are coming to my ETC. And so my NADH bind onto this first protein here, NADH, and it's going to leave the electrons, or the electrons bind onto that protein channel there, and becomes NAD+. Now this is good, because that can go where? Back to the glycolysis, back to links, back to Krebs, and be recycled and used again. That's how we recycle NAD+ in cellular respiration. Good. Now this electron is going to be driven down these proteins because there's an electron acceptor here at the end. What piece of cellular respiration have we not talked about, or what molecule do you think is my electron acceptor down there? At the end. What did we say is required for cellular respiration and we haven't used it yet? Oxygen. Yeah. Oxygen, we say, is my final electron acceptor. It takes those electrons and it's actually, we're going to make water from it. Okay? So oxygen's that final electron acceptor forming water. As these electrons move down these protein pumps, what's going to happen is it's going to be driving hydrogen ions into this inner membrane space. So I'm going to have hydrogen being pumped across these membranes, or pumped across that inner membrane as that electron flows down to that oxygen. Okay? This is called the electron transport chain. Okay? Now you might be wondering about FADH2. FADH2 just binds onto a different protein. And so it doesn't produce or pump as much hydrogen across. Okay, so NADH is a higher energy molecule than FADH2. NADH pumps more hydrogen across and can make more ATP. So now I have all this hydrogen in my inner membrane space, which means it's more concentrated here than in my matrix. So where does hydrogen want to go? Where does hydrogen want to go? Does it want to stay here? Do things like staying in high, rich environments? No. They want to go to the lower concentration. So my hydrogen wants to flow back through, and it does. Hydrogen flows back through this molecule that we call ATP synthase. And so, let me label that. Okay, my ATP synthase. It kind of acts like a wheel. And so as the hydrogen flows through, it spins, and it's going to take ADP and inorganic phosphate and make ATP from them. And per glucose, we are going to get about 28 ATP. I know if you do the math, it's about 3 per NADH, 2 per FADH2, and then we lose some along the way. And so that's why it doesn't come out exact. But it's about 28. Some books will say 30 ATP per glucose. So we call this last process where ATP is flowing through ATP synthase, we call that chemiosmosis. We're making that molecule with that movement of those chemicals. So if we come back here, we can say that our oxidative phosphorylation produces about 28 ATP. So per glucose, how many ATP molecules do we get? This is 28 to 30. So per glucose, how many ATPs do we produce? 32 to 34. Why is that advantageous compared to just doing aerobic respiration? Why is this whole cellular respiration process, why is that advantageous? Why is that good? It produces way more energy. In anaerobic respiration, or without oxygen, it only does glycolysis and only produces 2 ATP. This is why bacteria, they cannot get a lot of energy. They don't have this entire process. A lot of them use just glycolysis. And so we're more complex. We can have more energy. We can produce more energy through that cellular respiration process. So it's advantageous to be aerobic versus anaerobic in an evolutionary sense. All right, let's take a picture of the whiteboard. I'm going to erase it, and I'm going to do fermentation quick in the last 6 minutes. So if you need a picture of the board, take it now.
The first one's after cooling.
Because the batteries don't get enough battery space.
No, because I didn't have enough storage. But I don't need much stuff. So. Cleared up like 30 gigabytes.
All right. So in fermentation is what we do when we don't have oxygen, right? It's what bacteria do if they're anaerobes. It's what yeast do without oxygen. And why? They all start out with glycolysis. So everything starts out with glycolysis, which means that glycolysis has an evolutionary significance, right? If all organisms have it, it's a conserved process showing common ancestor. All right, so we're going to start out with glucose, and we're going to get 2 pyruvate. How much energy is produced in this process? How many ATP? 2. And we have NAD plus going to NADH. Now, can this process just stop here? And can this work forever? Could the process stop here and work forever? No, why? continue to produce pyruvate and NADH and get these 2 ATP. The goal is to make energy. I have 2 ATP.
There's no oxidative phosphorylation.
There's no oxidative phosphorylation. And what does oxidative phosphorylation do?
Removes the hydrogen off of there.
Replaces it back to NAD+. The entire goal of fermentation is so that we have more NAD+, so that can be cyclical and constantly being recycled inside the body, right? And so our pyruvate, if this was a plant and this was alcohol fermentation, our pyruvate is actually going to be broken down into a 2-carbon molecule, which means that what must be released? If it's a 2-carbon and pyruvate was 3, what has to be released? Carbon dioxide. Okay? And that 2-carbon molecule is going to oxidize NADH to NAD+, and it's going to produce 2-ethanol. Which is actually how we get alcohol in the fermentation process. So alcohol is made from plants, right? You take a plant-based product. In the case of wine, it's mainly grapes. In the case of beer, it's mainly wheat. In the case of other things, it might be barley, and it might be, when I was in Kenya, they would ferment baobab and all kinds of weird fruits. But that's how that happens. And if you've ever seen those processes happen, you can see the CO2 being released from that process to make that ethanol. In the case of animals, we undergo lactic acid fermentation. Bacteria can do, I'm not sure. Bacteria, I think, can do either one, but I'm not positive on that. Glucose, once again, to our 2-pyruvate, we get our ATP, NAD+, to NADH. Same problem, right? We need to somehow oxidize that back to NAD+, so it can be recycled or will run out. And so pyruvate is then going to be oxidized, NADH is going to be oxidized, pyruvate is going to be reduced to form a lactic acid molecule. 3 carbons still. We didn't lose carbon in this one. And this is how, like, cheese fermentation happens. This is how yogurt is produced using this process, removing that oxygen from the environment. And that's fermentation. For this one? No, just there's no oxygen present. And so when oxygen isn't present, then we can't go through all of this because oxidative phosphorylation can't accept those electrons. So fermentation happens without oxygen presence. And the whole goal is to replenish that NAD+. All right? Awesome. That's it.